Hello, my stellar students. It's time for another biology with Miss Judd. Now, we just completed our lab on gummy bears, and what we did was we put some gummy bear into some water and watched what would happen. Newsflash, our gummy bears grew so much. What? That's crazy. Who knew that would happen? Miss Judd knew that would happen. All of that water moved into our gummy bear. Now, as fun as it is to watch gummy bears grow so much, we can relate that to cells. Just like our gummy bear was different from the outside water, our cells are different to what's on the outside. And our cell membrane controls what moves in and out. It's that barrier between the cell and the outside world. So just like water moved into our gummy bear, our cell membrane controls what moves into our cell. So that's what our first lesson in Unit 3 is all about, that transport of material in and out of a cell. Now, the best person to tell us more about that is Miss Judd in the corner.
Hi, everybody. Miss Judd in the corner here to start us off with our first lesson in Unit 3 about cell transport. Now, the organelle that we're going to be talking about a lot here is about the cell membrane. Now, the cell membrane is super important because it controls what comes in and out of the cell. The cell membrane separates our cell from the outside world. It is made up of 3 parts. It is made up of phospholipids, proteins, and sugars. So it's not just the line that we sometimes draw, but it's made up of a bunch more things. There's a lot more going on here. Now, these membranes create a 2-layered structure, and it's constantly changing based on what the cell needs to come in and out. This is called being fluid. It's changing, it's moving. It also has a lot of stuff in it. That's called being mosaics. We have our sugars, we have our proteins, and we have our phospholipids. So let's dive into these pieces. Our phospholipids are made up of 2 parts. We have our hydrophilic heads. So these white circles here are the heads of the phospholipid. They love water. They want to be around water. They want to be exposed to water. Now, the yellow and orange here are the tails. Those push water away. Water is scary. They want to get rid of it. It is not great to be around. So my phospholipids have these 2 parts, the head that likes water and the tails that repel water. That's the main part of a cell membrane. Now, inside the cell membrane, we have our proteins and our sugars. Proteins facilitate the transport of things coming in and out. Sugars provide the structure. And we have 2 types of transport. We have active and we have passive, right? So that's what we're going to dive into next are these 2 types of transport, passive and active. Passive transport is movement into the cell with no energy whatsoever needed. So they move from a lot of molecules, so a lot of purple moves into the cell to where there's not a lot of molecules, from high to low concentration. Lots of purple molecules into not a whole lot of purple molecules. This passive transport is diffusion across a membrane using no energy whatsoever. Molecules just want to do it on their own. You can kind of think of molecules as being claustrophobic little things. They don't want to be around each other and they want to get away. So they're going to move as quickly as possible away from each other. Now, the words we use here to kind of define, we have concentration gradient. A concentration gradient is that there is a difference between the amount of stuff separated by my cell membrane. So in this image, I have a lot of blue dots on one side and not a lot of blue dots on the other, meaning I have a high concentration on one side and a low concentration on the other. This concentration gradient, meaning high to low. Diffusion is the actual movement. So my particles are moving from high to low concentration until they are equal. And we call this equal in this balance being at equilibrium, meaning that all parts of a space have the same amount of stuff in it. So we're going to define these words out. Another way to look at diffusion is the natural movement of high to low concentration. So we're going to watch a little clip here about the diffusion. Super simple, not a whole lot going on, but a simple diffusion is just moving from high concentration to low concentration. So lots of oxygen moving across the membrane until they're balanced. Remember, equilibrium is that balance. So what moves through passive transport? We have water, H2O. We've talked a lot about it. It's small, it's polar, and it moves in and out via osmosis. And we're going to dive into that. And any other really small molecule is going to passively just cross the cell membrane. Now, sometimes things are too big. They have a charge, they're too big. There's still a concentration gradient, high to low, but it's not just going to passively move. So what helps is proteins. We talked a little bit about proteins being embedded in the cell membrane. This is called facilitated passive transport. Still no energy, still just moving from high to low concentration, but now we're using these little green proteins to help move across because normally they would just get stuck. So these proteins help my molecules move. All right, so we talked a little bit about osmosis, water moving through, and how does that work? Osmosis is the diffusion of water to get to equilibrium. So water is still moving from high concentration to low concentration across a membrane until we get to equilibrium. So we see here water, and it's moving across the membrane. And if we look here, we have more water on this side, less water on this side. So we're moving from high concentration to low concentration. Now we have tonicity. Tonicity is just saying how much stuff is outside of the cell. So the relative concentration of stuff outside of the cell. And when there is more stuff on the outside, a higher concentration of particles on the outside, water will move out. Hypertonic solutions is saying my cell is inside of a solution that has more stuff on the outside. So water's going to leave. What this means in an animal cell is that it shrinks and shrivels. In a plant cell, because it has a cell wall, it shrinks and sticks to the cell wall. So you can kind of see what it looks like. In your notes, you can either write or draw this. A hypotonic solution means that this outside solution contains a lower concentration. So water moves into the cell. This means that my cell is going to expand and explode in an animal cell, or expand and hit the cell wall in a plant cell. In an isotonic solution, water moves in and out at balance. So I'm at equilibrium. Water's still moving, but it's balanced. So my cell looks the same. It just kind of stays the same. We've talked about passive transport. Now let's talk about the other type, active transport. Active transport talks about how things move in and out of the cell, but this time we need energy. So energy is required to bring things in and out of the cell. When with active transport, I want you to look at this image and note that there are a higher concentration here and a lower concentration here. My molecules are moving from low to high. That's opposite of what we were just talking about with passive transport. We are no longer trying to get away from each other. Instead, we're moving against the concentration gradient. So in active transport, we have proteins that help big molecules pass across the membrane when it's going against the concentration gradient. And off to the side on your notes, it might be helpful to write down that this uses energy. This is not something natural. Instead, energy is required to do this. So if we look, we have a high concentration of sodium, these little orange hexagons, and we're moving from low to high against the concentration gradient. Some of the ways this happens is with bulk transport. So when a cell needs food or a cell needs nutrients, it will engulf the food and pull it in. That's something that takes a lot of energy. That is called endocytosis. It's moving stuff actively into the cell, and it encloses around and pulls it in. The opposite of this is exocytosis. So when there's waste, it pushes stuff out of the cell. So these vesicles move and merge with the outside and pushes it out. Both of those require energy. This is important in our bodies. Just a little bit of relevance here. Why we're learning about this is because our brain neurons, our neurotransmitters, communicate with each other using this process. And when we dive into photosynthesis and cell respiration, we're going to talk about how things are moving across a membrane. Thanks, Ms. Judd in the corner. As always, great information. Now, we just learned a lot, so I'm going to send it to Ghost Ms. Judd to help us practice. All right, so here we're looking at our osmosis that we just talked about. In our directions, we're told to determine what type of solution the cell is placed in, write the name, and then draw arrows to show which way water is going to move. And then finally, we're going to describe what happens to the cell as a result of osmosis. So let's look at this first one. Now, what we want to pay attention to is how much water is on the outside and how much water is on the inside. And my water is this little dude right here. H2, 2 hydrogens, and an O. And I have higher concentrations of water on the outside and lower concentration of water on the inside. Remember that with osmosis, we are moving from high concentration to low concentration. So my water is going to move into my cell. Over time, what's going to happen is my cell is going to expand and explode. So we're going to write that. This type of solution, how I describe the fact that there's more water on the outside than on the inside, is called hypotonic. Hypotonic. Now, number 2, I have more water on the inside and less water on the outside. So my water is going to move from high concentration to low concentration. This is a hypertonic solution, and what's going to end up happening is my cell is going to shrink down because less water is on the inside. Lastly, I have the same amount of water on the inside than outside. So water is going to move in and out at the same rate. This is isotonic. The cell is going to stay the same. All right, on our second one, we're going to fill in the boxes using the word bank. So here I have my cell membrane, my fluid mosaic model with my phospholipids, my proteins, active and passive transport, and diffusion. So I'm going to number this because I am doing this virtually. You are going to actually write all those words and do that practice. So the first thing I want to look at is when we have energy, we know that's active transport. So this transport right here, because the energy is involved, is active transport. Whenever we don't have energy, whenever it's moving from high concentration to low concentration, that is passive transport. So all 3 of these are passive transports. Now, I remember I have 2 types of passive transports. I have those that have helpful proteins and those that don't. When there's a helpful protein, it's facilitated by that protein. So that's facilitated diffusion. When the molecule is small enough to just move across those phospholipids, that we just call diffusion. Great work, you guys. I am so proud of you, as I always am. I hope you have your notes completely handwritten by now. Don't forget to upload those onto Seesaw and check your checklist to make sure you have all your work done. You guys are great and make good choices.